When we talk about the electron transport chain in an animal cell, this process will occur in the mitochondria of a cell. Now, the process of producing ATP through the electron transport chain can actually be subdivided into two main categories. We have the actual electron transport chain, which consists of complex 1, 2, 3, 4, and oxygen as the final electron acceptor. And it includes the second category, which is the chemiosmosis. Chemiosmosis is the part where ATP synthase will pump the hydrogens through itself to produce ATP. Now, if we start from the very beginning, the electron transport chain will start in the mitochondria, and it's happening across the inner mitochondrial membrane. And we start off with complex 1. Now, complex 1 is approached by a reduced electron carrier known as NADH. NADH is carrying proton and electrons, and it will donate its electrons to complex 1, and it will release that hydrogen it's carrying as well. So when it releases those electrons to complex number 1, there are these specialized regions that we won't get into right now, but there's these specialized regions where these electrons will come to, and then they'll jump from one region to another until they finally bind to a protein in the cell membrane. As these electrons are jumping from one region to another region, this process releases energy. And that energy generates a conformational change within complex 1, which results in the pumping of hydrogens from the matrix of the mitochondria into the intermembrane space of the mitochondria. So why are we pumping hydrogens into that intermembrane space? Well, we want to create that electrochemical gradient. When we create that electrochemical gradient where we have more positive on one side and more negatively charged on one side, through this gradient, we can pump hydrogens, and as we pump hydrogens, we can carry inorganic phosphate through ATP synthase and create ATP. So in a way, this is a form of secondary active transport because we're building an electrochemical gradient, and then we're using that energy created by that electrochemical gradient to produce ATP. Now, so from complex number 1, we have NADH. It's oxidized to NAD+. And we lose those electrons, and those electrons get passed on to a protein over here, which is known as ubiquinone. Now, this ubiquinone will go on to complex number 3, and the ubiquinone is carrying those electrons. But what happens at complex number 2? Well, complex number 2 is similar to complex number 1 in that it receives this reduced electron carrier known as FADH2. FADH2 will be oxidized to FAD+, and it will drop off its electrons to complex number 2. But complex number 2 will not pump protons into the intermembrane space. It instead will just pass on its electrons to another ubiquinone molecule and pass it on to complex number 3. So to revisit, complex number 1 pumped hydrogens. Specifically, it pumped 4 hydrogens into the intermembrane space. Complex number 2 does not pump any hydrogens. And now we're going to complex number 3. Well, complex number 3 is receiving electrons from the protein carrier ubiquinone, and it's coming from complex number 2, and it's coming from complex number 1. And it's at complex number 3 now. When it's received by complex number 3, there's this other special molecule known as cytochrome C. Cytochrome C will, one by one, carry the electrons over to complex number 4. And once again, as these electrons are going from jumping from one area to another area, there is an energy release, and that energy release is used to pump hydrogens from the mitochondrial matrix to the intermembrane space. So once again, we see the pumping of 4 hydrogens. Now, once the cytochrome C transfers the electrons from complex number 3 to complex number 4, complex number 4 will also pump electrons to the intermembrane space, but it will only pump 2 protons into the intermembrane space. Now, at complex number 4, there's also an additional step that happens. So at complex number 4, we have an oxygen molecule. And oxygen is highly electronegative, so it has a high affinity for those electrons. And it will be the final electron acceptor. So the driving force of the electron transport chain essentially is oxygen. Oxygen will gain those electrons, and it will gain hydrogens from the mitochondrial matrix, and it will produce H2O. So starting from complex number 1, we pumped 4 hydrogens. Complex number 2 didn't pump anything. Complex 3 pumped 4 hydrogens, and complex 4 pumped 2 hydrogens. So we got this buildup of hydrogens on the intermembrane space. So the intermembrane space will become more positively charged in comparison to the mitochondrial matrix, which relative to the intermembrane space will be negatively charged. So now that we have this electrochemical gradient, we can utilize this energy to pump inorganic phosphate through ATP synthase. So ATP synthase will pump inorganic phosphate along with protons to create ATP. So essentially, we get ADP, we have inorganic phosphate, and we utilize that hydrogen to create ATP. And this is the chemiosmosis part of the electron transport chain. And that is essentially what happens. We have these four complexes. They all pump hydrogens except for complex 2. We receive electrons from NADH and FADH2, and we undergo a series of hopping reactions where an electron will hop from one area to another area, and that energy release is used to pump hydrogens into the intermembrane space. The final electron acceptor is oxygen. Oxygen will acquire those electrons and hydrogens to produce water. And the electrochemical gradient is utilized as energy to create ATP. Now, this process occurs in the mitochondria. It happens on the inner mitochondrial membrane, and specifically in the mitochondrial matrix and the intermembrane space. And this is just a basic understanding of what the electron transport chain is and how it functions.